Hey everybody. By this point, you've actually made it through 2 units. Pat yourselves on the back. We are going in this unit to get into all of the details that allow our cells to function. So we're going to look specifically at the components of our cells, specifically eukaryotic cells. So we're going to take a look at all of our organelles and all the basic structures that we find within cells here on Earth. This video is going to cover AP Biology topics 2.1 through 3, as well as 10 and 11. And it follows pretty closely with what is covered in the 4th chapter of the Biology in Focus textbook. So we've got a lot to get through in this video. So without further ado, let's jump into it. What we should hopefully remember from earlier biology classes or life science classes from middle school is that cells themselves are the most basic structural and functional units of life. This is one of the foundational principles of the cell theory. Now, all cells on Earth, that is prokaryotic cells and eukaryotic cells, they all share the same basic characteristics. That is that they are enclosed in what we call a plasma membrane. We'll look in the next video into the details of what makes up the plasma membrane, but it is primarily composed of phospholipids. They also contain within the plasma membrane cytosol. So cytosol is a semi-fluid suspension. You may hear it referred to as the cytoplasm in some cases, but it's this jelly-like substance in which all of the things within our cell are floating. They also contain chromosomes. So chromosomes being made up of DNA and associated proteins. So this allows us to store and transmit genetic information from one cell generation to the next. And associated with the DNA, they also contain ribosomes. Ribosomes are considered organelles, but they are not membrane-bound organelles. But their primary function is to produce proteins. So they translate the information that we store within the genome into functional units that can do all the odd jobs that our cells need in order to stay alive. Now, I used a couple of terms on the previous slide, notably prokaryotic and eukaryotic cells. These are our two classes of cells which we can differentiate based on the components that they have. And evolutionarily speaking, prokaryotic cells arose first, and then eukaryotic cells with this increase in complexity came later on. Now, our prokaryotic cells include our bacteria and our archaea, and they notably lack any sort of membrane-bound organelles. Essentially, they're like a one-room efficiency studio apartment. They do all of the work within that one structure. And so, lacking these membrane-bound organelles, it means that they do not have a nucleus. The amoeba sisters like to say pro rhymes with no, so no nucleus in our prokaryotic cells. And so our DNA is floating within this unbound, what we call a nucleoid region. It's like the predecessor to our nucleus. And oftentimes our prokaryotic cells are much smaller than our eukaryotic cells. Now, in contrast, our eukaryotic cells are our more complex cells. These include animals, plants, fungi, and protists, and they do have membrane-bound organelles to separate all of these different functions. And in this video, we're going to look at a lot of those organelles and the specific functions that they play within a cell. So you can think of organelles like organs in your body, where each has a role to play in keeping the cell functioning at optimum efficiency. Because they have these membrane-bound organelles, it means that we also have a membrane-bound organelle to store our DNA in. So DNA in our eukaryotic cells is contained within the nucleus, which itself is surrounded by a nuclear membrane. Now, U, as the Amoeba sisters will say, rhymes with true, so we have a true nucleus in our eukaryotic cells. And these cells do tend to be larger. Because we compartmentalize a lot of those functions, we can exist at a larger size. As mentioned previously, all of our cells, both prokaryotic and eukaryotic, are surrounded by what we call a plasma membrane. This is a selective barrier, which we term to be semi-permeable, so it allows the passage of some molecules, nutrients, and waste products that is in service of the cell's volume. Now, the general structure of our biological phospholipid bilayer here is, of course, phospholipids. Bilayer means that we have two layers of phospholipids, and we'll get into more in the next video the fluid mosaic structure of this membrane. But you can see here in this diagram that a lot of the parts of our plasma membrane are composed of different structures. It's kind of an array of different pieces. We have proteins, we have carbohydrate markers as side chains, and we will get into all of that lovely stuff more in the next video. Now, I mentioned when discussing the plasma membrane that it is related to the efficiency of material exchange from the inside of the cell to the outside of the cell, and vice versa. Now, when we talk about the metabolism of our cells, we're talking about what we take in and what we release from all of the different processes that are taking place within that structure. Now, metabolism, because it produces and needs particular inputs, has upper limits that depend on a critical ratio between the surface area of our cell and its volume. So we can see both in the math breakdown here and also in the graph to the right that as our cell size increases, the surface area increases proportional to a square of the linear dimension, while the volume increases proportional to a cube of the linear dimension. When we look at the actual equations for these values, we see where that square and where that cube comes from. But what this means is that the surface area does not grow as quickly as the volume does in terms of proportion. So that means that as our cells get larger, we run out of efficient surface area and we have too much volume. So we're able to make way too much waste product and it's not able to get out of our cell. And for that reason, cells tend to be small. They prioritize a high surface area to volume ratio, which means that that ratio is going to be a larger number. This optimizes the exchange of materials at our plasma membrane. And because having a low surface area to volume ratio decreases our efficiency overall. So we'll see some cells that have very unique structures, and we've hopefully established that structure helps us determine the function of something. And so having these unique longer or folded structures allows us to increase the surface area of our cell without increasing the volume to that same proportional degree. So we get a lot more bang for our buck in terms of efficiency of our more folded increase in surface area volume options here. So here I've got a little bit of a math practice for you guys. I assume that most everyone at some point has had some foundation in geometry. Say whether that is thinking back to finding surface area and volume in shapes in the actual high school geometry class or in middle school. But here is where it comes into play in biology. So we're going to focus on 4 different cell shapes in terms of geometry. We'll have cubes, rectangular prisms, cylindrical prisms, and spheres. And each one of those has a unique equation by which we can calculate its surface area and its volume. So here is where I'm going to ask you guys to practice with those equations and identify which of the cells here exhibits the greatest metabolic efficiency based on its measurements. And record your answer on your sheet. Another way that cells can effectively get around this idea of limited surface area to volume ratios is that they can divide their cell into smaller compartments, which we term to be organelles. Now, the majority of organelles that we are going to look at are membrane bound, so they are composed of the same sort of membrane that we find surrounding our cell. This compartmentalization increases our cell's efficiency without affecting the overall surface area to volume ratio because we are not limiting the surface area of the cell itself, just the internal structures. And so all of these different organelle membranes and our plasma membranes, they participate directly in our cell's metabolism, and they allow our eukaryotic cells to create much more products over a period of time than we would be able to produce in our basic prokaryotic cell. So what we are going to look at in the next portion of this video are all the organelles that I will expect you guys to recall for the AP Biology exam. Now, we are going to want to focus not only on the structure of these organelles, but also their function. What role do they play in the cell overall? We will start with the one that differentiates primarily our eukaryotic cells from our prokaryotic cells, that being the nucleus. So here's a figure from our textbook, and I've put the figures on a separate slide from the information that I'll go over so that you have an idea of the structure before we get into their function. So here, the nucleus is composed of a double-layered nuclear membrane, and we can see that that nuclear membrane is dotted with nuclear pores. Now, those pores, like windows into the nucleus, allow our RNA, which is our functional working copy of specific genes, to leave the nucleus, but they are not large enough to allow our DNA to leave the nucleus. So we house our DNA within this structure, keeping it safe, making sure that nothing bad happens to it, because if we destroy that DNA or if we disrupt it in some way, we can often have disastrous effects because it controls the information that makes all of our other products within the cell. Now, the nuclear membrane is continuous with some of our other membrane-bound organelles. Those membranes are connected. So you can also see in this figure that we have the rough endoplasmic reticulum, which we will cover a little bit more in the future. Also within our nucleus, we see that we have a nucleolus. The nucleolus is the site where we produce ribosomes, ribosomes being our sites of protein synthesis, which we'll also talk about a little bit more in the next few slides. So, what does the nucleus do? The nucleus contains most of our cell's genetic information. There is some genetic information that is stored in some other organelles, notably the mitochondria and the chloroplast, which hints at their prokaryotic origins. And it is enclosed within a double membrane nuclear envelope, which separates it from the cytoplasm and the rest of the cell. As mentioned previously, the nuclear pores, which dot that nuclear envelope, regulate the entry and exit of different molecules. And we have the synthesis of our ribosome components, the ribosomal RNA, within the nucleus. Looking next to what exactly we're talking about when we're talking about chromosomes, because this is something that we'll cover more in depth in later units, but it helps us to kind of understand the general structure when we're talking about chromosomes and DNA within the same general topic. So in the nucleus, our DNA is condensed into units called chromosomes. And different organisms have different numbers of chromosomes, different amounts of DNA. And each one of those chromosomes is a DNA molecule that is wound around associated proteins called histones. These are like beads and string. And together they form something called chromatin. So chromatin is this unwound version of a chromosome. And the majority of the time, our DNA exists as chromatin, wound around these associated proteins until we're ready to divide, when we condense into those chromosomes and we essentially make it easier for our DNA to be separated for cell division. Another big player looking at the basic structure of our nucleus and of our rough endoplasmic reticulum are our ribosomes. Now, ribosomes are organelles, but they are not membrane-bound organelles. Instead, they are organelles that are composed mostly of proteins, but also portions of ribosomal RNA that are the sites of our protein synthesis. So these are our protein factories. And our ribosomes can be found in two major locations within our cell. They can either be free-floating within our cytosol, or they can be bound to the outside of one of our types of endoplasmic reticulum. So that is the rough ER, where we find our ribosomes. Now, the type of protein that we produce really is what determines which ribosome is doing the building. Most of our ribosomes that are going outside of the cell will be made in our endoplasmic reticulum, and those that are being used inside the cell will be made within our cytosol. The ribosomes of eukaryotes differ slightly in structure. They're larger than those that we find in prokaryotes, which is another notable difference that will come up again when we discuss the origins of our mitochondria and our chloroplasts. The next major topic that we're going to talk about in terms of our organelles are the components of our endomembrane system. So endo meaning within. So it's our membrane system that's within our cell. And these are a series of membrane-bound compartments that are either continuous, which means that we are connected to each other, or we kind of get one product from one to another through transfer vesicles. So we're going to look at 6 major components, 2 of which we've already covered, those being the nuclear envelope and the plasma membrane. But we have yet to get into the endoplasmic reticulum, the Golgi apparatus, and the lysosome, and the vacuoles. So we're going to start with the one that is connected to the nucleus, that being our endoplasmic reticulum. So the endoplasmic reticulum is a series of membrane channels, highly folded, which means that we have a great increase in our surface area. And we have two forms. We have the rough, which is dotted with those bound ribosomes, allowing for the formation and production of proteins. And the smooth ER, which does not have any bound ribosomes, so we're not producing any proteins in our smooth ER. Instead, the smooth ER is primarily used to produce lipids and as a detoxifying organelle. So let's look first at the specific function of our rough and our smooth endoplasmic reticulum. As mentioned previously, the endoplasmic reticulum is continuous with the nuclear envelope. So they are attached, which allows for information to be more easily transferred from the nucleus to the endoplasmic reticulum. And it is making up a large majority of our cell. Almost half of the total membrane in our eukaryotic cells is found as part of our endoplasmic reticulum. And we have two distinct regions of our endoplasmic reticulum. We have the smooth endoplasmic reticulum, which lacks the ribosomes, which means that it does not make proteins. Instead, its primary function is to synthesize or make lipids. So this is where we create components of our phospholipid bilayer. It also metabolizes or breaks down carbohydrates. So for use in other processes, we may need to break down larger carbohydrates. And it is notable for detoxifying our drugs, poisons, alcohol, which is why we find a lot of smooth ER in liver cells. Also notably, the smooth ER stores calcium ions being very useful in some of our action potentials and in our neuron connections. Second, we have the rough endoplasmic reticulum, which does have the bound ribosomes littering its surface, which means that we produce proteins, primarily glycoproteins. The proteins produced by the rough endoplasmic reticulum can be distributed using transport vesicles, these being like little bubbles that surround our proteins and take them from one location to another. And we also produce some membranes within our rough endoplasmic reticulum. Next in our line of where do our proteins go once they are produced by our rough endoplasmic reticulum is the Golgi apparatus. I always think that the Golgi apparatus looks a little bit like a stack of flattened pancakes because we have these series of canals of membrane that take our proteins and move them through from one side to another. So we have two faces of our Golgi apparatus. We have the cis face, which receives the proteins from our endoplasmic reticulum, and the trans face, which releases those proteins in another vesicle to carry them from one location to another. The Golgi apparatus acts very much like a processing center. So if you can think like a distribution warehouse, that is our Golgi apparatus. We're attaching all of those things that we need to attach to proteins for them to arrive at their destination. So this is where we attach all of our carbohydrate chains to our proteins. So the Golgi apparatus itself consists of flattened membranous sacs, which we call cisternae. Cisternae, having those cis and trans faces, we also have a medial region in the middle. And the primary function of our Golgi apparatus is to modify the protein products coming from our endoplasmic reticulum. We manufacture certain macromolecules here, mostly carbohydrates, and we can sort, pack, and make them ready for transport in those vesicles. So this is the region of our cell where we are getting everything squared away and ready to move on to other areas. Not attached to the other parts of our endomembrane system, these are lonely organelles, which have a key function in our cell to break down materials. These are lysosomes, where the prefix lys means to break and some refers to body. So these are bodies of enzymes whose primary function are to break down different parts within our cell. So we call these enzymes hydrolytic. You may recall the term hydrolysis from the previous unit, which is the term we use to refer to the reaction by which we break down polymers into their monomer parts. So we actually break down our macromolecules within the lysosome. So we have a very acidic internal environment, very similar to your stomach, where we have all of those enzymes that work at a very low pH. We have the stomach acid. So this is the stomach of your cell. And the enzymes that are found within lysosomes can be utilized to perform two key roles within our cell, depending on the point in our life cycle that our cell is currently in. So the one that we deal with when our cell is going through normal life functions is phagocytosis, phago meaning eating, and cytosis referring to a cell process. So this is the process of cellular eating. And this is where we digest all of those food molecules. Now, this is just normal cell behavior. We have to break down what we take in order to use it in other chemical reactions. But let's say that our cell is old and it's worn out and we need to get rid of some of our cell parts. Well, the lysosome can break those down as well. This is termed autophagy. So this is the eating of one's self. This is where our cell recycles its organelles and its own macromolecules, and sometimes the entire cell itself, in order for us to have apoptosis in some cases, or regulated cell death. So here we have a couple of different depictions of our lysosome and its primary functions as phagocytosis and in autophagy. So I won't break these down anymore because I feel like a pretty good description onto what these actually entailed. But we see that actual process taking place within both of these diagrams. Lastly, as part of our endomembrane system, we have the vacuole. Now, a central vacuole is only found in our plant cells, and it helps to deal with turgor pressure within our plant cells because the central vacuole essentially acts like a big balloon. And this balloon can shrink and it can expand based on the materials that it has inside of it. Primarily, the vacuole is used to store water and dissolved ions for use in the cell. So, vacuoles are large vesicles, so they're large membrane bubbles derived from the endoplasmic reticulum and our Golgi apparatus. And they are utilized primarily for storage functions. So we look at different types of vacuoles based on what they're storing. So we can store food, which the food molecules are formed by our phagocytosis performed by our lysosome. They can act as contractile vacuoles, so we can be used to pump water in and out of cells, partially by controlling the osmotic pressure, which we'll talk about in the next video, within that vacuole region. And they can be storage vacuoles or central vacuoles that are used to store all of our inorganic ions like potassium and chlorine. Moving along from the endomembrane system, we have all of our organelles that are involved in energy or metabolism. So these are necessary for organisms to be able to transform the energy that they acquire from their surroundings. And we'll look at two of these in a little bit more detail in the next unit where we talk about photosynthesis and cellular respiration. But in eukaryotic cells, there are two primary metabolic organelles, those being the mitochondria and the chloroplast, that can convert energy into forms that cells can use for work. The mitochondria are our sites of cellular respiration, which is a metabolic process that uses oxygen to generate cellular energy in the form of ATP, or adenosine triphosphate. Mitochondria are found in both plant and animal cells. Anything that needs to create energy has mitochondria. Chloroplasts are found only in our plant cells and our other photoautotrophic organism cells. So these are sites of photosynthesis, which is a process that takes in light energy and uses it to generate carbohydrates. And finally, not to be outdone by the two heavy hitters, we also have peroxisomes, which are sites of oxidation. We will look at each of these in a little bit more detail now. The first of these metabolic organelles that we're going to look at is the mitochondria. The mitochondria being the site of cellular respiration, which allows cells to create ATP from different food molecules, is composed of two membranes. The outer membrane is more like that of the eukaryotic cell, but the inner membrane is more like that which we find in prokaryotic cells. Another key identifying piece of evidence that allows us to conclude that mitochondria were likely once free-living prokaryotic cells. The inner membrane of our mitochondria is highly folded, which we've established increases our surface area, allows us to be more metabolically efficient, very important when we need to produce a lot of energy for our cell. And so that inner membrane is folded into structures that we call cristae. And within that inner membrane, we have a fluid called the mitochondrial matrix. Each one of these regions will play more of a role when we discuss cellular respiration in detail. Another piece that you may find interesting and that allows cells to be more efficient overall is the ability for our mitochondria to both be separate organelles, but also to fuse together to create a mitochondrial network, allowing us to create a lot more energy in certain types of cells. Also notable is that our mitochondria have both their own DNA, which is contained as plasmids or circular rings, very similar to what we would find in prokaryotic cells, and their own ribosomes, which are also very similar in size and structure to our prokaryotic ribosomes. So mitochondria are found in nearly all eukaryotic cells. Of course, if a cell does not need to create large amounts of energy, they may lack a mitochondria, but they are primarily there to facilitate cellular respiration, aerobic respiration in this case. So they have that smooth outer membrane, a folded inner membrane made up of cristae, which creates two compartments within our mitochondria. We have an inner membrane space that separates our inner and our outer membrane. And within the inner membrane, we have that mitochondrial matrix. The folded structure of our mitochondria increases surface area, which allows us to synthesize more ATP overall. Looking to our other heavy hitter in terms of metabolic organelles, we have the chloroplast. Similar to our mitochondria, the chloroplast also has double membranes, the outer of which more similar to the eukaryotic cell, and the inner membrane more similar to our prokaryotic cells. Also, like the mitochondria, the inner membrane is highly folded. This time, we are folded into thylakoids, or coin-like structures that themselves are stacked into what we call grana, or stacks of coins. Now, in this chloroplast, we have a fluid suspension that is called the stroma. And the chloroplast additionally contains DNA like plasmids that are very similar to what we would find in prokaryotic cells, and their own ribosomes, very similar to those found in prokaryotic cells. So, also hinting at the fact that chloroplasts were once free-living prokaryotic cells. So chloroplasts are organelles that are found primarily in leaves and other green organs of plants and algae because they contain chlorophyll, chlorophyll being a green pigment which is used to absorb light energy. They also contain enzymes and other molecules that function overall in photosynthesis, all of which we are going to spend more than enough time in our next unit. And the chloroplast structure includes two main parts: thylakoids, which are our membranous sacs that are stacked into granum, and our stroma, which is that internal fluid. Lastly, we are going to just highlight here a lesser known metabolic organelle, those being the peroxisomes. Peroxisomes are primarily used for oxidation reactions in which they produce hydrogen peroxide and can convert it to water. Peroxisomes tend to be forgotten, I think, overall in terms of metabolic organelles. And of course, there are other organelles that we are not even touching on in this foray into cell structure. A lot of different organisms may have specialized organelles, but we are hitting the ones that most cells have. I've kind of been laying breadcrumbs throughout this entire piece, looking at all of the things that allow us to suggest or infer that mitochondria and chloroplasts were once their own free-living organisms. Now, this is what we call the endosymbiont theory. So endo meaning within, and symbiont referring to symbiosis, which in our ecology unit, we talked about a mutually beneficial relationship between two organisms as symbiosis. So we have three primary pieces of evidence to suggest that mitochondria and chloroplasts were once free-living prior to being engulfed by proto-eukaryotic cells and becoming their own organelles. That is, that they are enveloped by a double membrane, the outer membrane of which is more similar to that of the eukaryotic cell, and the inner membrane being more similar to that of a prokaryotic cell. That both mitochondria and chloroplasts contain ribosomes that are more similar in structure to prokaryotic cells than those that we find in the eukaryotic cell, and multiple circular DNA molecules called plastids. The fact that both chloroplasts and mitochondria have their own DNA molecules also allows them to duplicate themselves rather than being duplicated by the rest of the cell when it's ready for cell division. So they can grow and divide somewhat independently within cells. So you may be wondering at this point, how do materials move around in the cell? How do organelles stay where they are if they're suspended in this fluid? Well, that is thanks to the cytoskeleton, which is a network of fibers that extends throughout the cytosol and organizes all of the cell's structures and the activities going on. So we are controlling where things are located at what time, and when things need to move, that they have an opportunity to move. So the main functions of the cytoskeleton are that it helps to support the cell and maintain its shape, very similar to how your skeleton helps to support your body and maintain your shape. They also provide anchorage for our organelles and our molecules so that they stay relatively stable within that fluid. And this is what is actually moving the vesicles from one point to another. Motor proteins accomplish this through a very unique piece where they essentially walk along the fibers, a very unique method of transportation, to say the least. So we're going to look at some of the different fibers that make up the cytoskeleton. I do want to emphasize before we get into these that this is not something that you will need to memorize, just to be aware of the different structures that we have comprising our cytoskeleton within our cells. So there are three types of fibers that we're going to take a look at, and they vary in the material that they're made of, also in the thickness of the material itself. So we have microtubules. Microtubules are the thickest of our components. We have microfilaments, which are the thinnest of our components. These may also be termed actin filaments. They are made of actin. You find a lot of microfilaments in muscle tissues and muscle cells. And intermediate filaments, which, as the name suggests, have diameters that are somewhat in between microtubules and microfilaments. So looking a little bit more closely at microtubules, microtubules are made of tubulin, which is where we get that tubules part, so microtubules and tubulin. They are functioning to shape and support our cell, guide the movement of our organelles, but they also come up during cell division in order to separate our chromosomes. So these microtubules in animal cells grow from another organelle that is not membrane bound called a centrosome. The centrosome aids in the separation of our chromosomes, and so we will touch back on our microtubules when we get to cell division in Unit 4. They're also what controls our other motor or movement, locomotion based organelles. Again, not membrane bound, that being the cilia, our tiny hair-like structures, and the flagella, the tail that helps different cells move. Next, we have microfilaments. Microfilaments, remember, also called actin filaments, are built of actin subunits, and they function to bear tension, resist pulling forces, and to interact with another protein called myosin to allow cells to move once more. So microfilaments really show up a lot when we're talking about muscle cells, because those actin subunits are really what allow our muscles to contract as we move, and so very important. This is also one of the parts that may or may not get disrupted by certain changes in protein structure, because if we mess up our actin subunits or our myosin subunits, or our diskesin subunits, we can disrupt the function of our overall cell in terms of movement. And finally, we have our intermediate filaments. Intermediate filaments are, of course, those that are in a medium size. They're not the thickest, they're not the thinnest, and they're only found in some cells, but they primarily are used to reinforce shape and to reinforce the locations of our organelles. So only some cells have these. Other cells rely primarily on that microtubulin to perform the same role. So here is a diagram from the textbook that gets into a little bit more detail, shows some of the subunits that these are made out of and their varying diameters. And one more figure from the textbook. This is our two eukaryotic types of cells, plant cells and animal cells. These will be the ones that we use as the model cells. Of course, these have all of the organelles that we're talking about because they're models. Not all cells have all of the organelles that we've talked about today. Some of them have more of one than another. Some are lacking some altogether. That's because the type of organelles present in a cell determine the function of that cell. So if a cell plays a specific role in the body that only requires particular pieces, they will only have those organelles necessary for their function. This allows our cells to really get different jobs done, that is, they're specialized or differentiated between each other, and allows us as multicellular organisms to have specific roles for specific parts of our body. On that note, we will end the video here, everybody. Thanks for hanging in there, and hopefully you feel confident/comfortable with the organelles that we find within our eukaryotic cells, as well as the general structure of cells themselves. And I will catch you in the next video.